I want to spend a little bit of time talking about the two supporting connective tissues as an example of connective tissues overall. We're going to start with bone. Now, bone can be either compact, also known as cortical bone, or trabecular, also known as cancellous or spongy bone. Compact bone is a lot harder and more dense, whereas spongy bone tends to have a lattice-like arrangement that allows it to flex a little bit more. Since most bones contain both types of tissue, they actually get the best of both worlds here. We can see this if we look at the structure of flat bones, which are found primarily in the skull, and also at the long bones, which are the most common bone type inside of the body. In each of these cases, we see that the compact bone tends to make up the outer layers of the tissue, and the spongy bone is found deeper inside. Now, most of the examples that we're interested in of specific bones in this course are long bones, and so I'm really going to focus only on long bones from this point out, although pretty much everything that I say would apply to the flat bones as well. So, long bones get their name because they're longer than they are wide. The middle image on this slide shows the typical anatomy of a long bone. The shaft of a long bone is also known as the diaphysis, and it makes up most of the length of the bone. At either end of the bone, you have an epiphysis, which is typically where this bone would attach or articulate with another bone to form a joint. In between the epiphysis and the diaphysis is a transitional area of the bone known as the metaphysis, and we're actually going to talk about this quite a bit later on, and we'll refer to it as the growth plate. The rightmost image on this slide takes a look at some of the internal anatomy of the bone. So you can see that the outermost layer of the bone is actually a connective tissue wrapper known as the periosteum, and we're going to refer back to this a little bit later when we talk about bone development. Beneath the periosteum, we find the compact bone. Deep to that, we find the spongy bone. And deeper still, especially in the shaft of the bone, we'll find a hollow cavity which contains the bone marrow. This slide takes a closer look at the anatomical arrangement of the compact bone specifically. Now, compact bone is really like a system of tubes within a tube. It's similar to what you might get if you were to just grab a handful of straws. The system is known as the osteon-haversian system. So if we use the analogy that I just gave you, the handful of straws, then each straw would be known as an osteon. And at its hollow center, we would find a central canal, which contains blood vessels and nerves. And that makes bone a highly vascular tissue. In fact, that's one of the reasons why breaking bones is so incredibly painful. And if you break a larger bone like your femur, it's potentially life-threatening because of blood loss or a secondary infection. Each osteon is formed from bone cells known as osteocytes, which occupy spaces within a bone matrix known as the lacuna. As the bone matrix is deposited around those spaces, it forms this system of tubes that I've been talking about. Now, because the bone matrix is very dense, the osteocytes use a system of canals known as canaliculi in order to distribute nutrients from the central canals between the various layers of the tissue. Here, we're taking a closer look at the bone matrix under a microscope, and you can see that it's primarily composed of a protein called collagen. Those are the cylinders that you're seeing in this diagram, especially in the right-hand image. It's from the collagen that bone gets its flexibility. But the collagen fibers are actually studded with a crystal made from the mineral hydroxyapatite, which is composed of calcium and phosphate. The chemical formula is at the top of this slide. And because there's so much hydroxyapatite in the bone matrix, this is the primary storage site for calcium and phosphate inside of the body. But if you've watched any milk commercials recently, then you probably have already heard this before. Now, just a second ago, I was talking about the mature bone cells known as osteocytes. But keep in mind that connective tissues tend to have many different cell types within them, and so bone is no exception. There are at least 4 different cell types inside of a typical bone. If you take a look at the image here on the left, you can see that 3 of them share a common developmental pathway. The bone stem cells are known as osteoprogenitor cells, and in response to specific developmental cues, they'll mature into a cell called an osteoblast. Osteoblasts can then become osteocytes, and we've already talked about those. The 4th cell type inside of bone is known as an osteoclast, and it actually develops from a different ancestor, different type of progenitor cell known as a fibroblast instead. The osteoprogenitor cells are typically found near the periosteum, and they'll differentiate into the osteoblasts, and the osteoblasts are responsible for producing the bone matrix. I always remember this by remembering that the blasts are our builders. The mature osteocytes can also produce bone matrix, but they tend to serve more of a housekeeping function to try to maintain the overall integrity of the tissue. On the other hand, osteoclasts are primarily responsible for breaking down bone matrix. That process is known as bone resorption. And they do this by secreting hydrochloric acid, which can break down the hydroxyapatite crystal into its components, calcium and phosphate. Anybody that has good taste in TV, there's a famous scene from Breaking Bad where Jesse tries to get rid of a dead body by pouring hydrochloric acid on it in the bathtub. The science behind his idea was sound, so he must have paid good attention in A&P. But those of you that were fans of the show know that there are some other issues with his approach. Cartilage and bone actually have many similarities, and although cartilage is a little bit more flexible than bones, overall it's still a very, very strong tissue, and it has a pretty similar molecular structure as well. The similarities are also present in the names of the cell types in cartilage. So chondroblasts build cartilage, chondrocytes maintain it, and chondroclasts break it down. But just like bone, there are several varieties of cartilage as well, including hyaline cartilage, which is strong and flexible and found in places like your nose. Hyaline cartilage is also important because it's found at articulations or joints, and we're going to talk more about that later in the musculoskeletal system. Hyaline cartilage also makes up the embryonic skeleton during development as well. Fibrocartilage is arguably the strongest type of cartilage because we really only find it in a couple of places. We'll mention it later when we get to the spinal cord because it makes up intervertebral discs. Lastly, we have elastic cartilage. This is the most flexible type of cartilage, and you'll find it in places like your ear, which need to have both a well-defined structure. For example, the shape of your outer ear helps to funnel sound waves into your inner ear. But you also need to have flexibility, right? You don't want your ear to break off if you roll over in your sleep. So there are two models for cartilage growth inside the body. And in the first case, we're talking about interstitial growth, which is when cartilage grows in a place where there's already cartilage present. This is often described as growth from within the tissue. So this story starts off with the chondrocyte in its lacuna undergoing mitosis, and that leads to a situation where two chondrocytes occupy one lacuna. If you've ever shared a very small living space with someone, then you know that the situation is kind of untenable. So in the same way that your roommate probably left their stuff all over the place, each chondrocyte continuously deposits matrix into the lacuna. And as the matrix starts to accumulate in this interstitial fluid surrounding the cell, it eventually separates the two chondrocytes from each other. And we now have a situation where each chondrocyte has its own lacuna, and this whole process repeats after they go through mitosis. The second type of cartilage growth is known as appositional growth, and this is used to create cartilage de novo. In other words, to make cartilage in a place where there was no pre-existing cartilage, like during development. Now, this can also be used to repair damaged cartilage as well. So in this model, since there is no cartilage or the cartilage has been damaged, we actually have to differentiate from stem cells. So the stem cells differentiate and then form chondroblasts, which will start to secrete the matrix. And that's going to make the tissue start to take on the properties of cartilage overall. Eventually, once you have a cartilage matrix, the chondroblast becomes a chondrocyte, and it will continue to maintain and housekeep, and then follow up and transition into the interstitial model that we just talked about.